done with Miss Kalandada. Okay. So let's try to ask kung sino yung susunod.
Si Miss Sofia Ragundo po, sir.
Okay,
Sir,
sige.
ako po.
Okay, sige. You can now take the floor.
Naririnig niyo po ako?
Yes, dear. Loud and clear. Go.
Sorry. Okay, start na po.
Okay, sige.
Assalamualaikum and good morning, everyone. My name is Sofia Ragundo, and I'll be continuing the reporting session with another lesson. The first is the nucleus and the replication, and here are the objectives. So by the end of this session, we are expected to be able to describe the structure and features of the nuclear membrane, list the organization of the DNA molecule within the nucleus, and describe the process of DNA replication. The first one here is the nucleus. So on the previous lesson, we have learned the function of cytoplasm and its contents. Now we will focus on the much smaller parts, which is the nucleus. So belong pa rin siya sa cytoplasm. Nasa loob siya ng cytoplasm, but instead of focusing on the cytoplasm, we will go here with the nucleus and its contents. So the nucleus is the largest and most prominent of a cell's organelles. It is generally considered the control center of the cell. It stores all of the genetic instructions for manufacturing proteins. The nucleus is the controlling unit for every organelles, and it is located at the center of the cell. The reason behind nucleus being entitled as the master of all organelles is because it contains genetic materials. The nucleus controls and regulates the activities of the cell. Example is the growth and metabolism, and it also carries the genes, structures that contains the heredity and formation. So other cells, such as mammalian red blood cells or RBC, do not contain nuclei at all. So as I have said earlier, the nucleus is the control center of the cell. And dahil lang cells or lahat ng cells katulad ng RBC ay simple lang ang function, compared sa nucleus, mas mainam na wala ito or walang nucleus. The RBC just carry chemicals around, and it has the oxygen carrying capacity via hemoglobin. It also allows the cell to have its distinctive biconcave shape, which aids diffusion. So this shape would not be possible if the cell had a nucleus and a way. So without the nucleus, the lifespan of RBC is short, and so the body produces new ones constantly. So the red blood cells makes up almost half of our blood. The lifespan of a red blood cell is around 120 days, and lacking nucleus is a bit of a good thing for the RBC. So okay lang sa nucleus na walang RBC na walang nucleus kasi isa yun sa hindi na niya kailangan kasi kaya na naman niya mag-isa. So by the way, the body is producing new ones naman every time that the lifespan of RBC is ending. Next one is the skeletal muscle cell. A skeletal muscle cell contains many nuclei and is referred to as a multinucleated. These muscle cells are long and fibrous, often referred to as the muscle fibers. During development, many smaller cells fuse to form a mature muscle fiber, and the nuclei of the fused cell are conserved in the mature cell, thus imparting a multinucleated characteristics to mature muscle cells. So here is the photo of this muscle cell. Skeletal muscle cells are long and cylindrical. Cylindrical and striated. They are multinucleated, meaning that they have more than one nucleus. This is because they are formed from the fusion of embryonic myoblasts, and each nucleus regulates the metabolic requirement of the sarcoplasm around it. And skeletal muscle fibers, the nuclei, are distributed along the cell to maximize their internuclear substances. So as you can see here, makikita sa iba't ibang side nito ang maliliit na butil ng nuclei na nakasurround dito to maximize their internuclear distances. Next one is this one. This photo here is a figure of a red blood cell extruding its nucleus. Matured red blood cells lack a nucleus, so as they mature, erythroblasts extrude their nucleus, making room for more hemoglobin. The two panels here show an erythroblast before and after ejecting its nucleus, respectively. So losing the nucleus enables the red blood cell to contain more oxygen-carrying hemoglobin, thus enabling more oxygen to be transported in the blood and boosting our metabolism. This all situation right here, itong pag-extrude ng red blood cell sa nucleus, happened kasi mas mainam sa RBC na walang nucleus, lalo na sa matured RBC. Okay, we'll proceed here to the organization of the nucleus and DNA. The nucleus is surrounded by a membrane called the nuclear envelope. This membranous covering consists of two adjacent lipid bilayers with a thin fluid space in between them. So you can see here, wait lang. Itong dalawang parang lines, yan yung tinatawag nating nuclear envelope. And ang nuclear envelope ay isang parang barrier na nagsaseparate sa mga laman ng nucleus at sa iba pang parte ng cell to the cytoplasm. So itong laman ng nucleus, which is itong mga strands na maliliit, is nasa-separate by the protection of the nuclear envelope sa cytoplasm. It contains many important proteins and other substances, kaya ito yung nagsisilbing taga-protekta sa mga ito. Okay, next. Next one is the nuclear pore. So the nuclear pore can be seen, wait lang. Interception. So it can be seen here, itong parang flowers. It is called the nuclear pore. And the pores are responsible with allowing the exit and entrance of these substances. It also has a nucleoplasm that is different from the rest of the cytoplasm. Wala nangyari. Sorry. Next is the gel-like nucleoplasm. There are also nucleus, chromatin, condensed of DNA and associated proteins, and then nucleosome. So the nucleolus is here. Itong bilog dito sa gitna. Is the largest nuclear organism in eukaryotic cells. It is assembled around arrays of ribosomes specific as nuclear organizing regions are the sites of ribosomal DNA transcription. So the next is chromatin. You can see the chromatin here is in front, and makikita nito lilit na gitna ng nucleus, and we call it chromatin or chromatin fibers. So isa ang chromatin fibers sa pinaka-importanteng component ng nucleus dahil hindi lamang ito ang nagko-control sa genetic materials, kundi pati na rin sa pagkabuo ng chromosomes sa pamamagitan ng divide cells. So makikita ang chromosome bumuo dito sa isa pang unit na tinatawag nating genes. So sa loob ng chromosomes, nandoon yung genes. So the genes form the basis of heredity in all organisms. So these genes causes the similarities and differences of a certain organism with their parents. This functional unit of chromosome called genes are made up of molecules of DNA. This chromosome is only seen when the cell is about to divide, kaya kapag walang cell division na nangyayari sa loob ng nucleus, nakikita ang chromosome as single strands which can be called as chromatin instead of chromosome. Okay. So it is estimated that the humans have almost 22,000 genes distributed on 46 chromosomes. So, on this lower side, itong cisternae, wala dito nalagay, pero itong cisternae naman, isang function niya is the packaging, modification, and transport functions for the cell overall. Kumbaga, ang sister RNA ang nadalanan ng mga cell bago mapunta sa specific places nito. Halimbawa, protein and polysaccharides, ano nito, mapunta sa specified location nila. Outside nucleus is a plasma, and it is a solution that fills each cell and enclosed by the cell membrane. So, outside this are other organs like plasma reticulum, mitochondria, itong course. Next one is the DNA replication. The DNA replication is the copying DNA that occurs before there are a great deal of determination that was used in 1958 two scientists in California, namely So it is a process by which a DNA molecule is copied to produce two identical DNA molecules. Replication is an essential process because whenever a cell divides, the two daughter cells must contain the same genetic information or DNA as the parent cell. So, DNA replication is a biological process ng pagkakaroon ng dalawang replika ng DNA mula sa isang original DNA. Sa process na ito, ang strands ng isang DNA is nagsaseparate into two to form replicas. So, the four bases or DNA bases are adenine, thymine, cytosine. Their shape and charge, the two bases that compose pair always bond together. So, adenine always binds with thymine and cytosine always binds with guanine. So, ang palang pares ay base A, adenine and thymine, C, naman ang pair. Okay. I think yung apat na genus bases or nucleobases, ito na sila, makitang kapag pinagpapasa sila. So, the reason behind these pairs is para maintain sa pagitan ng dalawang strands ng DNA, also to maintain a proper stable structure of the parent DNA is the original DNA and the daughter DNA is formed by the process of DNA replication. So this strand here, you can see it here, ito dalawang strand ito. Here you can see na it ends with opposite numbers dahil nga opposite ang dalawang strand. So the other one is a 3' end, ito, prime end ito, and the other one has a 5' end, ito naman. They do not have the same direction as expected because that is how it works. It is because the main enzyme, which is DNA polymerase, can add nucleotides at the 3' end of the DNA. The replication can be with 5' end, 3' end because the new nucleotides can only be added at the 3' end. Kaya hindi pwedeng mag-connect itong strands. Ito, hindi siya pwedeng mag-connect dito. Hindi naman siya pwedeng maging 5 kasi dahil maaring hindi magkatugma ang bases and it wouldn't be complementary with each other. So, ito, masasagasaan ito and magkakabalik-baliktad na. And there are also 3 important groups in nucleotides, which are these 3, the nitrogenous bases, and then the sugar, and the phosphate. So, a phosphate backbone is a portion of the DNA double helix that provides structural support to the molecule. DNA consists of 2 strands that wind around each other like a twisted ladder. Each strand has a backbone made of alternating sugar or deoxyribose and phosphate groups. The deoxyribose apart from being a carrier for the 4 bases, which is the adenine, guanine, cytosine, and adenine, it is also the anchor for the phosphate, which sits then on the outside of the completed polymer. The phosphate moiety makes the final product, the DNA, an acid. and acid. Its chemical structures allow for the replication of cells in DNA's double helix configuration. So the nitrogen bases, on the other hand, is these 4 bases here. So tinatawag ito silang nitrogenous bases. And ito yung formula nila. Yes. The next one is the DNA replication. So there are 3 stages of DNA replication, namely stage 1, stage 2, elongation, and stage 3. So in stage 1, the 2 complementary strands are separated much like unzipping a zipper. Special enzymes including helicase untwist and separate the 2 strands of DNA. Then the stage 2, elongation, DNA polymerase brings in the correct bases to complement the template strand, synthesizing a new strand base by base. And then there is stage 3, which is the termination. Once the 2 original strands are bound to their own finished complementary strands, DNA replication is stopped and the 2 new identical DNA molecules are complete. Wait, there are photos here. Okay. May photo dito. Example lang, process ng DNA replication. So, the first one is the enzyme helicase will do process first. So, ito yung chromosome. And then, if you zoom it, ito siya, chromatin naman yung i-focus natin. So, ito chromatin. This is the original template. And then, kapag pumasok ang the helicase unzips the strands of DNA. Kumbaga, siya ang responsible sa pagkakahiwalay ng dalawang strands ng DNA. The helicase do it in a way that it breaks the oxygen or hydrogen bonds, between the base pairs. So kapag nahati niya na ito, iba't iba pang enzymes ang papasok dito to continue the process of replication. So the next one is the primer. Segment of RNA called a primer is then synthesized and acts as a starting point for the DNA synthesis. An enzyme called DNA polymerase next begins replicating the DNA by matching bases to the original strand. So this is the DNA polymerase. Ito din siya, dalawa sila. And the DNA polymerase performs the vital function of a new strands of DNA known as the daughter strand by matching and adding new nucleotides from the first strand. So take note that polymerase cannot initiate new transformation. So 2 strands expose single bases on each strand, and each exposed base has the potential to pair with nucleotides in solution. This new nucleotides came from a pole that has been chemically synthesized in the cytoplasm. So these free nucleotides here that is studied here is from the free floating nucleotides in your cell and are derived from the food you eat. After that process, the RNA primase will come, and like I said, that while polymerase cannot initiate strands, that is why primase came in help. So the enzyme primase has a short segment of RNA known as a primer, which acts as a guide for the DNA polymerase to synthesize this strand further. And then, in both cases, replication occurs so quickly because multiple polymerases can synthesize 2 new strands at the same time by using each one. The original DNA double helix as one of this original called leading strand, and then the lagging strand on the other hand. That are synthesized and as discrete short DNA fragments termed Okazaki fragments, which later form continuous complex DNA. So, Okazaki fragment begins with a primer DNA made by polymerase. So the Okazaki fragments are small sections of DNA that are formed during discontinuous synthesis of the lagging strand. They are important because they allow for both daughter strands to be synthesized, which are necessary for cell division. So once a synthesis is complete, the RNA primer are replaced with DNA, and one or any gaps between newly synthesized DNA segments are sealed together with enzymes. Oh my God, sorry. The DNA replication is crucial process. Therefore, to ensure that mistakes or mutations are reduced, the new synthesis of DNA. Once the DNA is in a cell is and divided into two cells, each of which has a identical copy of the original DNA. So kaya importante ang DNA replication ay dahil para i-ensure na lahat ng offsprings ay makakatanggap ng genetic information na katulad ng sa parent cell kasi kapag may error dito, maaaring mag-result yon into cancer. So the DNA replication ensures that each new cell receives its own copy of DNA. This mode of replication is the original DNA molecule is served in each new DNA molecule. This process continues until the cell's genome of an organism's DNA is replicated. It is very important that DNA replication place precisely so that new cells in the body contain the exact same genetic material as their parent cell. Protein synthesis, tapos na tayo sa DNA replication, then we'll proceed to protein synthesis. So by the end of this section, we are expected to be able to explain how genetic code stored within the DNA determines the protein form. Also describe the process of transcription, describe the process of translation, and discuss the functions of ribosomes. So protein synthesis is the process of creating protein molecules. In biological system, it involves translation. You might wonder how important protein is. In reality, it's very important because it's structure in transport, has enzymes, materials, and protecting the body. So proteins are large, complex molecules that play many critical roles in the body. They do most of the work in cells and are required for the structure, function, regulation of the body's tissues and organs. So photo here is a summary of the process. As you can see here, this is the original template of DNA, and when the transcription occurs, the DNA would look like this, the strands would look like this, and we are calling it RNA. So it would be the mRNA. So you can see many changes in here, such like the bases. As you can see, itong bases na thymine is ng uracil. So cells napalitan ng instead of having double strand is single stranded na ito kasi naging mRNA. So after the transcription, the translation will take place, and as shown here, the amino acids are formed. The amino acids are formed here. Ito sila. So we'll explain further the transcription and translation and the So next one is from DNA to RNA transcription. So this is the first process of protein synthesis, which is the transcription. So the gene expression begins with a cell called transcription, which is a synthesis of a strand of mRNA that is complementary to the gene of interest. This process is called transcription because the mRNA is like a transcript or copy of the gene's DNA code. So gene expression begins with a process called transcription. Tinatawag itong transcription dahil lang mRNA is parang transcript or copy ng gene's DNA code. Nagsisimula ang transcription na medyo pareha sa DNA replication, and ang kaibahan lang ay, di ba, sa DNA replication, ang two strands ng DNA is nagsaseparate talaga completely. Pero dito, kasi sa transcription, kaunting portion lang ng DNA ang pinaghihiwalay. Okay, sorry. So DNA is housed within the nucleus, and protein synthesis takes place in the cytoplasm. There must be some sort of intermediate messenger that leaves the nucleus and manages protein synthesis. Ang intermediate messenger na ito ay ang messenger RNA or mRNA. Ang single nucleic acid ang genetic code sa isang gene palabas sa nucleus para dalhin sa ribosome na kung saan nag-produce ng protein. Ang structure ng RNA at DNA ay may pagkasimilar pero may pagkakaiba rin. May itong photo dito. Ito, DNA and RNA. Unlike DNA kasi, ang RNA ay single-stranded and walang complementary strand. Ito, wala siya dito sa top. And then, the RNA's ribose sugar contains additional oxygen atom compared to DNA, and instead of base thymine, RNA contains base uracil. So, di ba sa DNA, ang base natin ay may base thymine na palaging kapares ng thymine. So, sa RNA, wala thymine, uracil. Kaya ang nangyayari is nagiging partner ng adenine is uracil kapag nasa RNA. Next is the stages. So as I've said earlier, parang similar itong transcription doon sa DNA replication kasi may tatlong stages din. Initiation, elongation, and termination. So the first one is initiation, a region of the beginning of the gene called the promoter, as a particular sequence of nucleotides trigger the start of transcription. And then the next one is the elongation. Transcription starts when RNA polymerase unwinds the DNA segment. One strand, referred to as the coding strand, becomes the template with the genes to be coded. The polymerase then aligns the correct nucleic acid with its complementary base on the coding strand. So after that, the termination will come. Polymerase has reached the end of the gene. One of the three bases are specific triplets code a stop signal which triggers the enzyme to terminate transcription and release the mRNA transcript. So ang mRNA or RNA polymerase will connect RNA bases to DNA. So mRNA is all around. It can go away from the nucleus to the ribosomes where proteins are made. The ribosome is made of rRNA or ribosomal RNA. I think dito. Wait. Ang reason kaya may mRNA is dahil ang DNA is masyadong malaki kaya hindi makita. Lang na lang makaalis sa transcription starts, diba? Ito ang DNA strands. And as you can see, masyado itong malaki para makalabas sa nucleus, so hindi siya pwedeng magamit ng buo. Pero kaya need man natin ng copy ng DNA para sa protein synthesis, kaya ang mangyayari is itong kaunting copy lang dito, ang ikakopy natin. And yun yung magiging mRNA natin. The process starts with an enzyme called RNA polymerase, as you can see here, and which binds to the DNA just before where the copy gene starts. Then sa unahan ng RNA polymerase, ang two strands ng DNA is nagsaseparate kaya lahat ng bases ng dalawang strand is naghihiwalay and na-expose yung bases nito. And then kapag ang strands ay naghiwalay na, ang RNA polymerase ay i-read lahat ng bases na na-expose and gagamitin yon to make an mRNA strand. So itong dito sa top na nadaanan ng RNA polymerase is may expose, and of course the nucleotides will make here another copy or RNA transcript. So ito na yung magiging transcript natin or mRNA natin na magagamit sa paggawa ng protein. So lahat ng bases sa mRNA at DNA ay pwedeng ipasa sa isa. Lang ay kapag sa RNA na, ang makakapartner ng adenine is uracil instead of thymine. After the RNA polymerase have made an mRNA, the DNA then closes. Ang lahat ng madadaanan ng RNA polymerase na strand ay tinatawag na template strand na ginagamit sa pagkakagawa ng mRNA. So after the transcription, hindi diretso agad na translation because dadaan pa sa spliceosome, sa process na spliceosome. So a spliceosome is a structure composed of various proteins and other molecules attached to the mRNA and splices or cuts out the non-coding regions. The removed segment of the transcript is called an intron. The remaining exons are pasted together, and an exon is a segment of RNA that remains after splicing. So this is the process of splicing. So as you can see here, ito yung buong DNA. So the spliceosome splices out the non-coding introns from the primary mRNA transcript and stitches the exons back together into the matured mRNA transcript. So this is the original look of the DNA. And then after the transcription takes place, ito na yung magiging result, which is the pre-mRNA. The pre-mRNA lacks structure that the messenger RNA requires. Thus, that is why we are needing the method of spliceosome to take care of the lacking structure. So itong green dito is called the exon, and the red one is intron. So intron and exons are nucleotide sequences within a gene. Introns are removed by RNA splicing as RNA matures, meaning that they are not expressed in the final messenger RNA, while exons go to the mRNA. Ibig sabihin, ang intron at exon ay nag-i-exist sa DNA kasi nucleotide sequence nila, but kapag pumasok na ang transcription, doon na nangyayari ang splicing o yung pagkatagpi-tagpi ng mga exons, tapos doon na rin inaalis yung exons para magawa yung DNA. And then, kapag nagawa na ang mRNA, nandun lang is yung exons, pero yung introns ay naiiwan dito sa pre-mRNA. So, bakit nga ba hindi sinasama ang introns sa mRNA? Simple because not only the introns do not carry information to build a protein, they actually have to be removed in order for the mRNA to encode a protein with the right sequence. So kapag ang spliceosome ay pumalya sa pag-alis ng intron bago mabuo ang mRNA with the extra junk in it will be made, and the wrong protein will be produced during transcription. So the process of removing introns and reconnecting exons is simply called splicing. The next one is the RNA to protein, which is translation. So this is the last part of synthesizing a protein. And then translation is the process of synthesizing a chain of amino acid called a polypeptide. Translation requires two major aids. First is a translator, the molecule that will conduct the translation, and second, a substrate on which the mRNA strand is translated into a new protein, like the translator's disc. Both of these requirements are fulfilled by other types of RNA, and the substrate each translation takes place in the ribosome. So before we start the process of translation, we have to remember that ang lahat ng 3 bases ng DNA at RNA, like TAG, CAG, and ATG sa DNA, G2, CCA, and UCG sa mRNA, which is called triplets or mostly known as codon codes for a specific acid, we should have to familiarize that. Though makikita ito dito sa sunod na slide. So, we should know that every making of proteins in our body kailangan ng 20 different amino acids, and lahat ng amino acids na yon is may iba't ibang different base codon. So to start the process, ang ribosome at mRNA is nababind together and is ready for the ribosome to start building proteins by adding one amino acid at a time. So by the way, the ribosomal RNA is a type of RNA that together with proteins composes the structure of the ribosome, and ribosomes exist in the cytoplasm as two distinct components, a small and a large subunit. So a ribosome functions as a micro machine for making proteins. Ribosomes are composed of special proteins and nucleic acid. The translation of information and the linking of amino acid are at the heart of the protein production process, which the process takes place in the ribosome. So next is a transfer RNA. Transfer RNA is a type of RNA that ferries the appropriate corresponding amino acid to the ribosome and attaches each new amino acid to the last, building the polypeptide chain one by one. Thus, tRNA transfers specific amino acid from the cytoplasm to a growing polypeptide. The sequence of 3 bases on the tRNA molecule is called the anticodon. So ang amino acids ay dinadala sa ribosome sa pamamagitan ng tRNA or the transfer RNA. Ang itsura ng tRNA is may amino acid sa top nito and anticodon naman sa bottom. Ito ang whole process. So ito ang transfer RNA. So ang anticodon nadala ng RNA complementary sa bases dito. Ito ang mRNA dito. So kasi ito ang cytoplasm, itong bilog dito. And as you can see here, masyadong malaki itong DNA. And because kailangan natin ng kopya ng gene sa paggawa nitong protein, so instead of getting all this and ilabas natin, ang kinukuha lang natin is yung kopya nito at para ilabas. And by the way, mRNA is all around, so free siya, kahit siya dito pumunta, is pwede siya. Kaya siya yung ginagamit natin kasi mas madali siyang ginagamit ng body natin kasi siya yung madaling makalabas sa nucleus. So, ang tatlong bases sa mRNA is ito, and then ito ay nagko-code sa amino acid na dala ng tRNA. Halimbawa, ang dalang codon ng anticodon ng tRNA is UCE, uracil, cytosine, and adenine. And then ipapartner ito sa first three bases ng mRNA, which is, let's say, adenine, guanine, and uracil. And ang code na ito is magre-result into amino acid na serine. So kapag ang tRNA ay nakapagdala ng tamang order ng amino acid, the ribosome can start building up a chain of amino acids with the chains. When the chains is bind together, it folds up on itself and make a protein. But if the protein synthesis goes wrong, diseases such as cancer can occur. And yun lang po. Sir? Tapos na po. Wala siya dito?
Nawala ata si sir.
Ay na siguro ang net ni sir.
Ay, doon na miya kinagaw ko niya.
Wala may kailangan.
Ako din sa recorded guys. Ako ang grade. Ako pre-recorded. Ay, recorded na.
Ng process.
Hintayin na lang natin.
Tayin muna natin.
Mag-continue na daw, nawala.
Continue na daw. Continue? Sige. Sige, go, Makala.
Yan lang to.
I-end mo muna yung share screen mo, Bismillah.
Hi, good morning. Hi there, everyone.
Hello, sir.
Okay,
Yes, sir.
tapos na si Ragundo.
Yes, sir.
Okay, sige. Wala yung kuryente saglit, no? Okay, sige, go. So this time we have Sahara Makala ulit.
Visible po
Is
yung
this
screen
part
ko,
of
sir?
group 1?
Yes, sir.
Yes, Sahara.
Ay, wait. So, good morning to each and everyone. I'm Sahara Makala, and I will be discussing about cell growth and cell division. By the end of this section, You will be able to describe the stages of the cell cycle, discuss how the cell cycle is regulated, describe the implication of losing control over the cell cycle, and describe the stages of mitosis and cytokinesis in order. So before we proceed to the process of cell growth and cell division, let us first know what is somatic cell. I guess most of you are familiar with the term somatic cell, right? A somatic cell is a generated term for a body cell, and all human cells are somatic cell except for the cell that produces eggs and sperm, which is what we call reproductive cell. So, that's why ang somatic cell, this is also called a building blocks of our body. Within a somatic cell, we can find there the homologous pair of chromosome, which is the 2 copies of a single chromosome. Since we have 23 homologous pair of chromosome in each of the somatic cell, we are called a diploid organism, and the condition of having pairs of chromosome is known as diploidy. Kung merong life cycle, meron din tinatawag na cell cycle. The cell cycle is the sequence of events in the life of the cell from the moment it is created at the end of the previous cycle of cell division until it then divides itself, generating 2 new cells. So kung merong isang cell, ang cell na yon will go through the process of cell division to form a new cell. At ang new cell naman na yon will pagdadaanan niya rin yung pinagdaanan ng previous cell na pinanggalingan niya. There are 2 general phases in cell cycle. First is the interphase, which is the period of the cell cycle during which the cell is not dividing. So in this phase, this is where primarily have cell growth occur, but not cell division. The majority of cell in the interphase, the majority of cell are in interphase most of the time. The interphase are divided into 3 distinct phases. The first phase is the G1 phase, which is the first gap or growth phase in the cell cycle. Here, all the organelles and cytoplasmic components replicate. Sa S phase naman or synthesis phase, this is the period during which a cell replicates its DNA. Kung sa G1 phase, yung mga organelles and cytoplasmic component ang nare-replicate, dito naman sa S phase, the one which replicates is the DNA. Then, the G2 phase is the second gap phase during which the cell continues to grow and makes the necessary preparation for mitosis. So here, all enzymes needed to aid in the process of cell division are produced. In interphase, most eukaryotic cells spend a great deal time here. So this is the structure of chromosomes. Itong dalawang pair na to, itong red at saka blue, yan ang tinatawag na homologous chromosomes. Tapos, ito naman yung centromere. Ito. This is the structure that attaches one sister chromatid to another. And sa region ng centromere, makikita mo dyan yung kinetochore, which is itong dalawang bilog. Yan, isa yung large protein complex that assembles in a specialized region of the chromosomes called centromere. The next phase is the mitosis and cytokinesis. So during this phase, a cell undergoes 2 major processes. First is the mitosis. Mitosis is divided into 4 major stages that takes place after interphase. First is the prophase, during which the loosely packed chromatin coils and condenses into visible chromosomes. Then metaphase. During this stage, the sister chromatids with their attached microtubules line up along a linear plane in the middle of the cell. In anaphase, it takes place over a few minutes. Then when the pair of sister chromatids are separated from one another, forming individual chromosomes once again. In telophase, this is characterized by the formation of two new daughter nuclei at either end of dividing cell. So meron akong picture dito ng process of mitosis. Sa prophase, as you can see, there are chromosomes condense and becomes visible. Ang makikita mo, itong bilog, yan yung tinatawag na nuclear envelope at nasa loob niyan yung tinatawag nating chromosomes. Spindle fibers emerge from the centromere. So itong parang may mga thread-like. Yan yung tinatawag na spindle fibers. Then, here in the prophase, nuclear down. So itong nuclear envelope, it will break down at mas magiging visible sa atin yung tinatawag na chromosomes once na ma-breakdown yan. Then, centrosomes move toward opposite poles. In prometaphase, chromosomes continue to condense. So, you will see also here na yung mga chromosomes, they are attached to kinetochores. Tapos yung kinetochores appear at the centromeres. So nagsilabasan yung centromere ang kinetochore sa centromere. And metaphase naman, kung dito sa prometaphase hindi pa masyadong align yung chromosomes, dito nag-line up siya, naging align na siya, naging straight na siya. Before siya mag-proceed sa anaphase, kailangan naka-line up yan. Kasi kapag nag-proceed agad siya sa anaphase na hindi pa siya naka-line up, magkakaroon ng abnormalities now which can lead to certain diseases. Then each sister chromatid is attached to a spindle fiber originating from opposite pole. So itong sister chromatid, they are attached to both spindle fibers. Unlike dito sa metaphase, tigi-isa lang ang naka-attach sa kanila na spindle fiber. Dito sa metaphase, both spindle fibers from the opposite poles are attached to the sister chromatids. In the anaphase, dito na sila pinaghiwalay, so wala talagang forever. Sister chromatids, now called chromosomes, are pulled toward opposite poles, so pinaghiwalay na sila. Then, certain spindle fiber begin to elongate the cell. In the telophase naman, yung mga sister chromatids na pinaghiwalay, they are surrounded by nuclear envelope, itong parang bilog. So hindi sila masyadong visible because they are inside the nuclear envelope. Then the mitotic spindle fiber breaks down, unti-unting magdi-disappear ang spindle fibers sa telophase. And then, the spindle fiber continue to push poles apart. Sa cytokinesis naman, this is the process. The process is then followed by cytokinesis, which is the dividing of cytoplasm and cell body into two new cells. So, dito sa cytokinesis, meron na tayo ditong two new daughter cells, and they are divided, meron separated sila ng naka-separated sila ng cleavage furrow. Let's proceed to cell cycle control. Mechanism of cell cycle control. A checkpoint is a point in the cell cycle at which the cycle can be signaled to move forward or stop. So, in the mechanism of cell cycle control, meron tayong tinatawag na checkpoint kung saan siya ang nagsisignal kung magpo-proceed ba yung cell sa another phase or mag-stop. Once na hindi pa prepared ang cell, it will not proceed to another phase kasi maaaring magkaroon ng homeostatic imbalance kapag nag-move forward siya pero hindi pa siya masyadong prepared. At each of these checkpoints, different varieties of molecules provide the stop or go signals depending on certain condition within the cell. We have this what we call a cyclin, which is one of the primary classes of cell cycle control molecules. So together with cyclin, we have this what we call cyclin-dependent kinase or CDK, which is a group of molecules that work together with cyclins to determine progression past cell checkpoints. So this is the picture of how mechanism of cell cycle control. Sa G1 phase or sa kung saan cell growth occurs, makikita natin dito yung G1 checkpoint. So before maka-proceed ang cell sa next phase sa S synthesis, dadaan muna siya sa G1 checkpoint. At once na hindi pa siya prepare, the G1 checkpoint will signal to stop the cell. But once it is prepared, it will allow the cell to go through the next phase. The cyclin D at saka tong CDK4 at saka cyclin E together with the CDK4, they are working together para maging maganda yung takbo ng cell growth dito. Then, ang pangalawang checkpoint is the G2 checkpoint. Nasa G2 phase siya makikita. Before maka-proceed ang cell sa next phase, it will prepare first the cell so that it can proceed to the another phase. At the third checkpoint naman, which is the M checkpoint or metaphase checkpoint, dyan, it prepares the cell in metaphase. Kailangan naka-align, yung sinabi ko kanina, kailangan naka-align yung chromosomes before siya ma-split into two. Kasi once na hindi pa siya naka-align, tas na-split siya into two, maaaring magkaroon ng abnormalities which can lead to certain diseases. The cell cycle out of control implications. Most people understand that cancer or tumors are caused by abnormal cells that multiply continuously. So, kapag mawalan ng control ang cell cycle, maaring magka-cancer ang isang tao kasi, of course, the cell will, kapag nawalan ng control yung cell cycle, the cell will multiply continuously. Just imagine, nagde-drive ka ng car tapos wala siyang brake. Di ba, there's high possibility na mabangga ka or mapahamak ka. At worst, baka ikamatay mo yun. Just like in the cell cycle, kailangan meron siyang alam niya kung kailan siya mag-stop or at kailan siya mag-go to another phase. If the abnormal cells continue to divide and stop, they can damage the tissues around them, spread to another parts of the body, and eventually result in death. So that's the worst case. It can result in death. Failures of control may be caused by inherited genetic abnormalities that compromise the function of certain stop and go signals. Environmental insult that damages DNA can also cause dysfunction on those signals. So maaring hindi mag-function normally ang signal na yun once there's an environmental insult, just like example of this environmental insult are for example, your environment are contaminated, possible siya na maka-damage sa signals na yun sa mga checkpoint. Often a combination of both genetic predisposition and environmental factors led to cancer. Cancer arises from homeostatic imbalances. So, dito na lalabas yung importance ng homeostatic balance kasi yung cell cycle control is also a mechanism of is also a homeostatic mechanism. That's why precise regulation of the cell cycle is critical for maintaining the health of an organism. As we all know, cancer is an extremely complex condition capable of arising from a wide variety of genetic and environmental causes. Typically, mutations and aberration in a cell DNA that compromise normal cell cycle control systems led to cancerous tumors. However, we have what we call proto-oncogenes and tumor suppressor genes which maintain homeostatic balance. So, kailangan you have proto-oncogenes and tumor suppressor genes. Otherwise, there might be homeostatic imbalance in the cell cycle process. Let's proceed now to the cellular differentiation. By the end of this section, you will be able to discuss how the generalized cells of a developing embryo or the stem cells of an adult organism become more differentiated into specialized cells. Distinguish between the categories of stem cells. What is stem cells? A stem cell is an unspecialized cell that can divide without limit as needed and can, under specific condition, which is differentiated into specialized cells. Stem cells are divided into several categories according to their potential to differentiate. First is the totipotent. These have the potential to differentiate into any of the cells needed to enable an organism to grow and develop. So this can also give rise to a new organism. The example for this is the fertilized egg or zygote. Next for that is pluripotent. Pluripotent has the potential to differentiate into any type of human tissue but cannot support the full development of an organism. So this also has the capacity to renew by dividing, and the Example for this is the embryonic stem cells. Next for that is the multipotent, which has the capacity, which has the potential to differentiate into different types of cell within a given cell lineage or small number of lineages, such as red blood cells or white blood cell. Finally, multipotent become further specialized, and oligopotent cell will be created. Oligopotent is limited to becoming one of a few different cell types. The example for this is the lymphoid or myeloid stem cells. The unipotent is fully specialized and can only reproduce to generate more of its own specific cell type. So this is capable of developing into only one mature cell type. The example for this includes spermatogonia, which can become only sperm, and oogonia, which can only become eggs. Differentiation. All cells contains, wait lang, nangyari dito. When a cell differentiates, it may undertake major changes in its size, shape, metabolic activities, and overall function. So how do the different cell types come to be different, considering that cells in the body, beginning with the fertilized egg, contain the same DNA? So just like in a movie script, merong iba't ibang actor na they share the same script. However, they only reading their own part of the script. Similarly, sa cell din, it contain the same DNA, but each type of cell read the portion of DNA that are relevant to each function. So in biology, this is referred to as the unique genetic expression of each cell. In order for a cell to differentiate into its specialized form and function, it need only manipulate those genes, and thus those proteins that will be expressed and not those that will remain silent. The primary mechanism by which genes are turned on or off is through transcription factors. A transcription factor is one of the class of proteins that bind to specific genes on the DNA molecule and either promote or inhibit their transcription. Let's proceed to stem cell research. So, stem cell research aims to find ways to use stem cells to regenerate and repair cellular damage. As we all know, over time, most adult cells undergo the wear and tear of aging and lose their ability to divide and repair themselves. The mechanism that induce a non-differentiated cell to become a specialized are poorly understood. And in that's why in a laboratory setting, it is possible to induce stem cell to differentiate into specialized cells by changing the physical and chemical condition of growth. Also, researchers have recently developed induced pluripotent stem cell from mouse and sorry. Also, several sources of stem cells are used experimentally and are classified according to their origin and potential for differentiation. Because of the capacity of stem cell to divide and differentiate into specialized cell, it offers a potential treatment for diseases such as diabetes and heart diseases. So that is the importance of stem cell research. Yung mga sakit na dati walang treatment, because of stem cell research, nagkaroon sila ng treatment for those diseases such as the diabetes and heart diseases. Then, researchers have recently developed induced pluripotent stem cell from mouse and human adult stem cells, and these cells are genetically reprogrammed multipotent adult cell that function like embryonic stem cell. So they are capable of generating cells characteristic of all three germ layer. Ang main point dito sa stem cell research, this research was conducted to find ways to use stem cells to regenerate and repair cellular damage. I think that's all. Thank you.
Okay. I think we don't have time na, no, for the Q&A. Perhaps yung question and answer would be next meeting na. So which is on Tuesday. Okay? Is that okay,
Yes,
everyone?
sir.
Yes, sir. Yun lang po.
Okay.
Yes, sir.
Yes, okay. But before we end, pa-stop share muna ako, Miss Sahara. Okay, tapos na ba yung group 1? Ito na ba yung lahat sa group 1?
Yes po, sir.
Okay, congratulations, group 1. So
Yes, sir.
again, Tuesday we will be having group 2, and then before we will proceed to group 2, there will be question and answer muna for Ragundo, and then of course for Sahara. Okay, but before that, before we will end the session, I would like to share the template of our synthesis. Yung sa synthesis template niyo for journal. Nakikita?
Wala, sir. Ito na, sir.
Okay. Sige. Okay, so this is now our research journal article synthesis. Okay, so bali, isusulat niyo dito kung ano yung module topic. So yung topic na doon sa syllabus. So since tapos na tayo sa discussion ko nung una, so you will write an introduction to the human body. Dito na banda. And then, since tapos na rin tayo for group 1, for the next synthesis naman, lalagyan na naman dito ng the cellular level of organization. And then this one, for the title, this is the title of the journal. And then yung author, si sulat. And then yung year published. And then yung complete URL kung saan nyo nakuha yung journal. Okay, klaro?
Yes, sir.
Sir.
Okay, and then you have to read the article first for you to write your introduction. Naka-slash yan. So you have to give us the introduction about it, and then the context, and then the rationale. Actually, kung ano yung purpose ng research, makikita nyo naman doon. No? Again, when you write your synthesis, you will not write everything. Mura lang mag-copy-paste sa journal. Please do not do that. Claro? Klaro
Yes,
ba?
sir.
O, baka ang mangyari
Yes, sir.
dito, mag-copy-paste lang tayo na wala ng purpose na nag-synthesize tayo. So, and then of course, the methodology. So you have to write the methodology. The whole methodology, please give us the understanding. Parang nag-summarize lang kayo nito. And the results and findings. So you have to look for the results and findings were in. Hindi na yung kailangan yung mga there are research articles kasi na ang dami nilang results and findings. All you have to find ay yung pinaka-importante. And then of course, the conclusion, nilagay ko ng recommendations kasi meron ding mga journals na meron silang recommendations. Kung wala naman, so okay lang. Okay? Klaro?
Yes, sir.
Yes, sir.
Okay, so I will right after this session, I will upload the template. And then, Sir, paano ba namin gagawin? Again, I have provided you some links sa Google Classroom natin, mga suggested na links para doon kayo makakuha ng journal article. Okay?
Sir,
Sige.
any journal
Walang
article?
questions? Yes. Any journal article related to the topic. No? Tingnan nyo lang, yung sa akin naman, yung sa Introduction to the Human Body, para siyang general ng human anatomy and physiology. So ano yung magiging hint niyo? Lahat ng kukunin niyo ay based lang sa topic na na-discuss. Now, like for example, sa akin, ang dami kong na-mention doon. I mentioned about anatomic directions, for example. So ano yung mahanap ninyo na nandun sa topic ko, within the topic. So hindi naman lahat ng topics na-discuss ko doon ay kailangan yung kunan ng journal article, isa lang. So kahit saan doon. The same with the sa cellular level of organization na topic sa group 1. Ang dami nilang na-discuss, they discuss about cell protein synthesis, they discuss about active transport, passive transport, ang dami. So hanap lang kayo doon kung saan kayo magfo-focus. Okay?
Yes, sir.
Nakuha ba? Everybody, na-gets ba?
Sir,
Na-gets
kibali
natin?
po.
Yes. Ayan.
Sir, bawat reporter po, sir, gawan po namin ng journal synthesis.
Ano? Bawat topic. Bawat topic lang. Not a reporter ha. Bawat major topic.
Okay po, sir.
Okay, so like for example, yung sa akin na report na discuss, yung titingnan nyo lang yung sa syllabus. Di ba yung akin is Introduction to the Human Body. So isang journal article for the Introduction to the Human Body, isang journal article for Group 1, yung cellular level of organization.
Bali,
Okay?
sir, sa isang group po, sir, na isang synthesis, sir?
Journal article.
Ay, journal article.
Yes, isa lang. Yes, isa lang.
Thank you, sir.
Okay, very struggling kung every reporter isa yung isang article every reporter. Ang pangit din siya, mapangit din kung every reporter kasi nag-overlap yung discussion niya. For example, si Kalandada nag-mention siya ng parts of the cell, parang na-stop yun kasi some discussion ng parts of the cell ay kukontinue ni Ragundo, for example, so ba overlapping. So hindi natin ma-identify kung kanino talaga tayo magagawa ng journal article. That is why per group, isa lang yung journal article. Meaning, in a very major topic, isa lang yung article na isi-synthesize. Pili lang kayo kung saang topic doon. Sa major topic. Nakuha?
Yes, sir.
Sir.
Okay, klaro? Klaro
Yes,
ba sa
sir.
lahat? Okay, sige.
Opo,
So I think
sir.
we are done. Okay. So I think we are done. I will now upload the synthesis template so that makahanap na kayo. Okay, so if you don't have any questions, thank you for coming, everybody. I wish you have a good day, no, for the rest of the day. So good luck, everyone. God bless. Keep safe always.
Thank you, sir. Bye, sir.
Bye bye.
Thank you po.
Bye. See you on Tuesday. Thank you.